Okay, this video is on protein synthesis, part 2 on translation. So if you haven't watched part 1 on transcription, you should probably do that first. So it has the same learning outcomes, understanding here all the different parts of transcription and translation, how they go together to synthesize or make a protein. So we're looking at the translation part of it now. So we've already made a messenger RNA. And so the main part of what happens here is that that mRNA will attach to a ribosome, which will help it to translate the information in the mRNA into a polypeptide, an amino acid polymer, which then folds into a protein. So in eukaryotic cells, what we're talking about here is the mRNA is made in the nucleus, travels out to the cytoplasm where it meets these ribosomes here in green, and that makes the polypeptides. In the eukaryotic cell, obviously it's all happening in the cytoplasm, and the mRNA comes out from being next to the DNA, again attaches ribosomes, and away we go. So what are the main things going on here? Well, really what we're doing is we're trying to make an amino acid chain or a polypeptide. In order to do that, there are 20 different amino acids that need to be added together in the right order in order to make the right protein. And so the mRNA provides the information as to which amino acids go in what order. The transfer RNAs bring those amino acids in, and the ribosome coordinates the whole thing. And there are different tRNAs for each amino acid. So this one with the yellow one has what we call a codon, an anticodon on it, which matches the codon on the mRNA, and those two things work together, the anticodon and the codon, in order to make sure that the next tRNA fits this sequence or is complementary to this sequence, and the right amino acid gets added on. And remember, when we were talking about RNAs, we talked about 3 different types of RNA. Well, here they are. The messenger RNA is here. The transfer RNA, the tRNA, is there carrying the amino acid. And within this ribosome, there are proteins and ribosomal RNA which help do this whole process. Now, although the diagram's drawn like this for simplicity, what you should be aware of is that there'll be multiple ribosomes attached to each mRNA. And in fact, here is a transmission electron micrograph, a TEM, with an electron microscope, showing here's bacterial DNA. There's mRNA being made off that, and as soon as that mRNA is made, ribosomes are attached to it. And in fact, here you can see lots of ribosomes attached to this piece of mRNA, and the little polypeptide chains coming off them. And this is the diagrammatic version of that. So we're making proteins all the way along. The important question is then, how is the information in the mRNA translated into the language of amino acids in order to make the polypeptide chain? And this is shown here in this diagram. So the RNA will have a sequence of letters, bases. And remember, we have C, G, U, and A in RNA. This gets divided up into 3-letter blocks that we call codons. So GCU, ACG, GAG, these are all codons. And these will be unique and provide information for the amino acid that they translate to. Now, it took scientists quite a lot of time to work out what this coding was. So you can imagine it's like coding from one language to another, where you have a sequence of letters that becomes a sequence of words in a different language. And that's why the process is called translation, and the coding is referred to as the genetic code. So here there are different ways of representing this. Sometimes it's wheels, sometimes it's tables, but here's our understanding of the genetic code. So with 3 letters, what you can do is the first letter has 4 options. So if you only had a 1-letter code, you've got 4 options to go with. A 2-letter code will give you 4 times 4, 16 options, and a 3-letter code will give you 4 times 4 times 4, or 64 options. So there are 64 different variations of your 3-letter code, but only 20 amino acids plus some punctuation to put in there. So sometimes it's called redundant, that we have extra codons that code for the same amino acid. So let's look at a letter sequence here. So if it's U, the first letter is U, so we go into this row. The second letter is U, so we go into this row. And then the third letter is U, and that's this one here. UUC will also give us phenylalanine. UUA and UUG give us leucine. These four also give you leucine, so there's 6 different ways to get leucine, so that's pretty good. This one here is really important. The codon AUG gives you methionine, which is what we call the start codon. So whenever you have an AUG, that tells the ribosome we're going to start a protein now. We're going to start making a polypeptide. We put methionine on the end, and that's the beginning one. These ones are valine, proline, all the different amino acids. These ones here, UAA, UAG, and UGA, are called the stop codon. So at this point, if that mRNA's come through and we have the codon UAG, that tells the ribosome we are stopping the polypeptide now and we're not going to add any more amino acids, and the polypeptide is released ready for folding. Folding is the next video. So polypeptides are made by the mRNA gives the information, the tRNAs bring the amino acids, and the ribosome puts it all together. It's really good for you to have a practice of going through this on your own, and so this online activity gives the information of how to do that. There's also some other videos that you can look at, but please do the online activity where you have to do the transcription and translation yourself. It's the best way to understand the process.